Good evening, everyone. I welcome you all to the second live session on bioinformatics algorithms and applications course. So, my name is Ramakrishna. So, I'm a PhD scholar working under Professor Michael Cromio, IIT Madras. So, coming to the week one session. So, before moving on to the week two session, I would like to know how many of you have attended the previous Monday session. So, that helps me. You can find the polls option where the question stated, have you attended the previous Monday session or not? You can answer yes or no. Yeah, if someone are unable to access the polls, they can use the chat box for their answers. Okay. So let me, for those who have not attended, so let me tell about brief about you the session. The session is be interactive and where the response of the students is also important and it will be collected using Google Meet polls, if not chat box, or students are free to open their mic and speak. And finally, if you have any questions and you wanted to answer at the last, you can use the Q&A option, which I'll be starting in few minutes. And you can also see the polls option under this triangle, square, circle mark. You can find an option polls where all the questions posted by me will be displayed. So coming to the session, in the initial part of the session, I'll be providing summary of the week 2 lectures or whatever the week that corresponds. Next, I'll be taking up the sample problems and solving along with you and providing you the answers. And if time permits, I'll be trying to solve the questions from the portal as well as I like to take the questions from the students. So before moving forward, I have few more general questions. So take up the question number 2 and 3 in the polls. So where whether you guys have watched the week 2 lectures and solved the week 2 assignment or not. I expect most of you to solve the week 2 assignment as the deadline is over. So we have this small communication and date clashes, so we'll be solving very soon, and where the sample solving sessions will be happen before your assignment deadline over after here. Okay, I see some people didn't watch the videos. Okay. Okay, let me start with the week 2 session. So coming to the summary. So the first lecture discusses what are micromolecules. By definition, micromolecules are very large molecules that are formed by the polymerization of small molecules called monomers. Coming to the term molecule refers to very large molecules or something that consists of more than one atom. In 1920, Hermann Staudinger coined this word, where macromolecules are so huge that these are made up of more than 10,000 or more atoms. And sometimes macromolecules are termed as polymers also, since they are formed by the polymerization of molecules such as carbon, hydrogen, oxygen, and nitrogen also. The monomeric units of these macromolecules are polar in nature with their tails and heads or with different physical and chemical properties. Coming to the types of macromolecules, there are four main types of biological macromolecules according to mammalian system. They are carbohydrates, nucleic acids, proteins, and lipids. Although these are classified separately in different segments of course, The principles that govern organization of these three-dimensional structures are the same for all of them. Therefore, we could consider them together. Coming to the brief explanation of carbohydrates, carbohydrates are nothing but polymers of carbon, hydrogen, and oxygen, and they can be classified as monosaccharides, disaccharides, and polysaccharides. So, carbohydrates are also found in starch, fruits, vegetables, milk, and sugars, and they are important source of healthy diet. Second is nucleic acids, and nucleic acids were also discussed in the problem solving session of week 1. So, nucleic acids include DNA and RNA, which are the polymers of nucleotides. Can anyone answer through chat box what is the difference between DNA and RNA in terms of nucleotides? Again, I'm repeating the question. Can anyone answer what is the difference between RNA and DNA in terms of nucleotides? Yes. Yeah, I see some answers coming up. Okay. Yeah, the major difference is the timing and uracil. So, coming to the explanation of nucleotides, nucleotides comprise a pentose group, a phosphate group, and a nitrogenous base group. So, all the hereditary information is stored in DNA, and DNA is synthesized into RNA and proteins. Coming to the proteins, proteins are considered as major biomacromolecules, and they are polymers of amino acids, which include carboxylic and amino group. Finally, lipids. Lipids are the hydrophobic set of macromolecules, that is, they do not dissolve in water or polar solvents. So these involve triglycerides, carotenoids, phospholipids, and sternoids. They help in the formation of a cell membrane, formation of hormones, and in has stored as fuel. So here, next lecture deals about proteins and their properties. As I mentioned before, proteins are polymers of amino acids. Amino acids are nothing but the organic compounds containing the basic amino group, that is NH2 amine group and COH carboxylic group. The ingredients present in the protein are amino acids. Both peptides or a sequence of amino acids or proteins are long chain of amino acids. Although there are 20 amino acids which are involved in construction of a protein. Out of 20 amino acids, our body can easily synthesize a few of its own. Since our body can synthesize on its own, they are called as non-essential amino acids. These include alanine, asparagine, arginine, aspartic acid, glutamic acid, cysteine, proline, and some others. Apart from these, there are 9 amino acids which are essential as they cannot be synthesized by our body. These are called essential amino acids, include isoleucine, histidine, lysine, leucine, and some others. Coming to the structure of acid amino group, amino acid have an amino group, carboxylic group, and a hydrogen bonded to the same carbon. They differ from each other in their side chain group called R. So this side chain R gives rise to 20 different types of amino acids. That is, the four groups are amino acid has a the four groups that are attached to the C alpha are a amino group, that is NH2, COOH group, carboxylic group, a hydrogen atom, and a side chain R. Further, these amino acids can be classified according to their properties. The important factors are charge, hydrophilicity, size, and functional groups. So these properties influence protein structure and protein-protein interactions as well. For example, water-soluble proteins tend to have hydrophobic residues such as leucine, isoleucine, valine, phenylalanine, tryptophan buried in the middle of the protein, whereas hydrophilic chains are exposed to the aqueous solvent. Coming to the other class of proteins called membrane proteins or transmembrane proteins, tend to have outer rings, outer exposed rings of hydrophobic amino acids that helps in anchoring them into the lipid bilayer. And some other peripheral membrane proteins have patch of hydrophobic amino acids on the surface that sticks them to the. So based on their environment, these hydrophobic or hydrophilic amino acids tend to play crucial role in protein functions as well as protein stability. In similar fashion, proteins that bind to positively charged molecules have surfaces rich in negatively charged amino acids. From the image, can anyone tell what are the negatively charged amino acids? Yes, D and D, that's correct. Thanks for your response. Yeah, so conversely, while proteins binding to negatively charged molecules have surfaces rich in positively charged amino acids like glycine and arginine. And coming to the other properties of amino acids, some amino acids have special properties, that is cysteine. Cysteine can form covalent disulfide bonds to other cysteine residues, and proline forms a cycle to the polypeptide backbone, and glycine is more flexible than other amines. That is why it tends to absorb glycine more in turns. Coming to glycine and protein are strongly present within the low complexity regions of both eukaryotic and prokaryotic proteins. So let's shift our topic towards the protein structure. Protein structure, as I mentioned, proteins are one of the most abundant organic molecules in the living system and have the most diverse range of functions of all macromolecules. Proteins may be structural, regulatory, contractile, or protective, as they may serve in transport, storage, or membranes. Each cell in a living system may contain thousands of different proteins, each with unique function. Their structures, like their functions, vary greatly. But however, we know proteins are nothing but a polymer of amino acids which are arranged in a linear. Polymer is nothing but some chain or some order of arrangement of these amino acids, 20 different amino acids. Coming to the process of synthesizing, which has displayed in the image, the process of synthesizing a protein from mRNA template is known as translation, and mRNA is loaded onto the ribosome and is read in 3 nucleotides at a time. So, can anyone tell me what is this 3 nucleotide set called as? We have discussed this already in previous section. So, how the mRNA is made it before translated, it is read as a 3 sets, like 3 nucleotides at a time. So, what is this set of 3 nucleotides called? Triplet codon, yeah. Yes, they are called as codons. And in general, proteins are always biosynthesized from N terminus to C terminus. The size of the synthesized protein can be measured by the number of amino acids it contains. One second. Yeah, the size of a synthesized protein can be measured by the number of amino acids it contains and by its total molecular mass. The sequence and number of amino acids ultimately determine the protein's shape, size, and function. Each amino acid is attached to the other amino acid by a covalent bond known as a peptide bond, which is formed by a dehydration reaction, that is, loss of water molecule. The carboxylic group of one amino acid an amino group of second amino acid combined releasing a water molecule. This results in a peptide bond formation. The products formed by such linkage are called polypeptides. While the terms polypeptide and proteins are sometimes used interchangeably, a polypeptide is technically a polymer of amino acids, whereas the term protein is used for a polypeptide or a polypeptides that have combined together and which have a shape and unique function. Most of the proteins fold into unique 3D structures, and the shape into which protein naturally fold is known as a native state conformation. Although many proteins can fold unassisted simply through the chemical properties of their amino acids, others require aid of molecular chaperones to fold to their native states. Biochemists often refer to four distinct aspects of protein structure. They are nothing but primary structure, secondary structure, tertiary structure, and a quaternary structure. Primary structure is the sequence of amino acids or a chain of amino acid. Secondary structure is regular repeating of local structures that are stabilized by hydrogen bonds. The most common examples are alpha helix, beta sheets, and turns. Because secondary structures are local, many regions of different secondary structures can be present in the same protein molecule. Next comes in the order is tertiary structure. Tertiary structure is gives the overall shape of the single protein molecule, that is the spatial relationship of secondary structures to one another. Tertiary structure is generally stabilized by non-local interactions, mostly by the formation of an hydrophobic core or hydrophobic collapse, and also through salt bridges, hydrogen bonds, disulfide bonds, and even post-transitional modifications. And the secondary structure gives local regions, and once this tertiary structure is formed with these secondary structure elements is what controls the basic function of the protein. The final thing is quaternary structure. The structure formed by several protein molecules or several polypeptide chains, which are called protein subunits or subunits, which function as a single protein complex. Coming to the different functional classes or classifications of the protein. Proteins can be informally divided into three main classes, which correlate with the tertiary structures, that is globular proteins, fibrous proteins, and membrane proteins. Okay, I'll start discussions with a globular protein. Almost all globular proteins are soluble and many are enzymes. The best known role of the protein in the cell is as enzymes, which catalyzes the chemical reaction. So the major role of enzymes is acts as a catalyst in reactions. And these enzymes are usually highly specific and accelerate only or few, they are specific to some chemical reactions. And they carry out most of the reactions involved in metabolism as well as manipulating DNA in process such as DNA replication, DNA repair, and transcription. Some enzymes act on other proteins to add or remove chemical groups in a process known as post-translational modifications. The molecules bound and acted upon by enzymes are called as substrates. And enzymes are nothing but they are also proteins that consist of hundreds of amino acids. It is still, enzymes are large number of amino acids. It is usually known a small fraction of residues that come in contact with the substrate that are directly involved in catalysis. The region of the enzyme that binds the substrate and contains the catalytic residues is known as any ideas? Again, I'm repeating the question. The region of the enzyme that binds the substrate and contains the catalytic residues is known as. So there is a particular where this enzyme and the substrate binding happens. So there's a particular name for that site.
Active site.
Yes, active region or active site, that's correct. Yeah, so this active site on an average has 3 to 4 residues. Next, class of proteins are fibrous proteins. So fibrous proteins are structural proteins such as collagen, which is the major component of connective tissue, or keratin, the protein component of hair and nails. Third class is membrane proteins, which the major role of membrane proteins is to serve as receptors or provide channels for polar or charged molecules to pass through the cell membrane. These membrane proteins act as receptors whose main function is to bind a signaling molecule and induce or start a biochemical response in the cell. Many receptors have binding site exposed on the cell surface and an effector domain within the cell. That is, these are two parts where the exposed site recognizes the signal molecule and the effector domain within the cell, which starts an enzymatic activity inside. The other name for this membrane proteins, they are also called as transmembrane proteins, since they span across the membrane region. And they also serve for the transport, molecular transport. So this molecular transport, the major role of these transport proteins or membrane proteins is to alter the permeability of the cell membrane so that the small molecules or ions, other proteins can pass through them. That is, they create an exchange in between the external and intracellular environments. So the way which happens are called as ion channels or pumps, so which allows the molecules to enter or exit the cell. So many ion channel proteins, for example, are specialized to select only for a particular ion. For example, you must have seen potassium channel or sodium channel often discriminate only for one of the two ions. Next is, what is the topic of bioinformatics is sequence alignment. In bioinformatics, sequence alignment is way of arranging the sequences of DNA, RNA or protein to identify regions of similarity that may be a consequence of functional, structural or evolutionary relation between the sequence. So our main aim of sequence alignment is to find or separate out the regions of similarity between given sequences, or what is the sequence of dissimilarity among them. To compare two or more sequences, like alignment is nothing but a comparison. So how we are comparing by arranging them in a particular order or aligning them in a particular order. So to compare them, it is necessary to align the conserved and unconserved residues across all the sequences. That is nothing but we are trying to find out where the regions of insertions or deletions must have occurred since after the divergence from the common ancestor. So when the sequences are aligned, it is possible to identify the locations of insertions or deletions since there isn't some divergence like match or mismatch criteria. So there are three possibilities that can arise once a sequences, two or more sequences are aligned. That is, all the bases match. So complete match means that there is no change since their divergence. The bases mismatch. The second criteria is mismatch. This means that there is a substitution since their divergence, and there is base in one sequence and no base in other sequence. That there is insertion or deletion since their divergence. So alignment of these sequences in bioinformatics serves as a number of purposes. That is, DNA sequence data can be aligned to assemble larger contiguous DNA sequences, whereas proteins can be aligned to identify the important regions, such as I mentioned, active sites. So these are the most conserved part in proteins. And DNA sequence can also be aligned to identify the regulatory control regions. So when we align only two sequences, it is called as a pairwise sequence alignments. So pairwise sequence alignment methods are used to find the best matching piece of alignments of two query sequences. Query is nothing but input sequences. So as I mentioned, pairwise alignments can only be used between two sequences at a time, but there are efficient ways to calculate and are often used methods that do not require extreme precision to perform a pairwise alignment. The three primary methods for producing a pairwise alignments are dot matrix, dynamic programming, and word methods. Okay, I'll be discussing the three primary methods one after the other. So let me start with the dot matrix. The dot matrix approach, which implicitly produces a family of alignments for individual sequence regions, and is a qualitative or conceptually simple, but it has its own limitations of time consuming to analyze on a large scale data. The image here is shows a dot plot between the two sequences. So the topmost one is sequence 1 and the leftmost one is the sequence 2. So using the dot matrix, it can be easy to visualize and identify certain sequence features such as insertions, deletions, repeats, inverted repeats. Okay, now we'll see how to construct a dot matrix. To construct a dot matrix, the two sequences are written along the top of the row and leftmost column as shown here. And wherever you see a point of match, a dot is placed. And this is the other name for this plot is also called as a recurrence plot. So what happens if I try to plot a dot plot for a very closely related or very highly similar sequences? Can anyone answer? So what happens, or where can I see the points if I try to plot a dot plot for very closely related sequences or very highly similar sequences? So how can we see the alignment of or position of these dots? We see most of the we see a straight line along the diagonal as shown here without any gaps when we try to align them. That is, the dot plots of very close related sequences will appear as a single line along the matrix main diagonal. Diagonal is nothing but joining one corner to the other corner. The problem with the dot plots as information displays include noise, lack of clarity, non-intuitiveness, difficulty in extracting the matches summary or statistics as the sequence size increases. Still, apart from these limitations, dot plots can also be used to assess repetitiveness in single sequence by a sequence can be plotted against itself, and regions that share significant similarity will appear as a lines of the main diagonal. This effect occurs when a protein consists of multiple similar structural domains. Second is dynamic programming. So before going into the dynamic programming, let's study about causes for sequence dissimilarities. The first major cause. Okay, let me ask you guys first, what could be the reasons for dissimilarity when we align a protein, or what, like how do we mention the similar sequence? Why? What happens? Yes, mutations, correct. Okay, I'll start with it. The causes for the sequence dissimilarity, the first one is mutation, where a nucleotide or an amino acid at a certain position is replaced by the other one. So it's a replacement term. Secondly is insertion. At a certain location or before a nucleotide or before amino acid, a new nucleotide or amino acid is inserted, which causes an insertion or increase in the length also. Second thing is deletion. The quite opposite term of insertion is a deletion. Next is indel, insertion or deletion happening at a time. Coming to the technique of dynamic programming, the technique of dynamic programming can be applied to produce global alignments via Needleman and Wunsch algorithm. and local elements via Smith-Waterman algorithm. In typical usage, protein alignments use a substitution matrix to assign scores to the amino acid matches or mismatches, and a gap penalty for matching an amino acid in one sequence and to introduce a gap in the other alignments. So in practice, when we see how this match score, mismatch score is simply assign, when we are performing, assign a positive match score, a negative mismatch score, and a negative gap score to get a proper or a good alignment. As I mentioned, pairwise sequence alignments methods are concerned with finding the best matching piece of uh, this global and local alignments have their own advantage or own information production in there. So generally, the purpose of these is to find the homologous or relative of a gene or a gene product in a database of known example. So the information that we get from these alignments is useful for answering variety of biological questions such as the identification of sequence of an unknown structure or a function, or to study the molecular evolution. Okay, let me discuss about pairwise global alignment. So the global alignment between two sequences is an alignment in which all the characters in both sequences participate in the alignment. So we are not removing or avoiding any of the letters that are present in the sequences. And these global alignments are useful mostly for finding closely related sequence or to judge whether the given two sequences are closely related or not. As these sequences are also easily identified by the local alignment methods, the global alignment is now somewhat deprecated as a technique. Further, this also has several complications to molecular evolution such as domain shuffling, which prevent these methods from being. So global alignment fails to recognize the closely related sequences when there is a domain shuffling happening. So next method is the local alignment method. So these local alignment methods tries to overcome the limitations of the global alignment method by finding the related regions within the sequence. For example, if we take a positions 20 to 40 of a sequence A might be aligned with the positions 50 to 70 of a sequence B, and this doesn't care aligning all the sequence at a time. So these are local regions, alignment of local regions. And this method is more flexible than global alignment and has the advantage that relates the regions which appear in different order on two proteins. Next is word methods or pairwise heuristic local alignments. These word methods are also known as k-tuple methods, which are heuristic methods and are not guaranteed to find an optimal alignment solution, but are significantly more efficient than dynamic programming. So these word methods are especially useful in large-scale database searches where it is understood that large proportion of the candidate sequences will have essentially no significant match with the query sequence. Word methods are best known for their implementation in the database search tools FASTA and BLAST, where the word methods identifies a series of short, non-overlapping sequences in the query sequence that are then matched to the candidate database sequences. The relative positions of the word in two sequences being compared and are subtracted to obtain an offset. This offset will indicate a region of alignment if multiple distinct words produce the same offset. So only if this region is detected, these methods apply more sensitive alignment criteria and thus many unnecessary comparisons with the sequence of no appreciable similarities are eliminated. Okay, with this, coming to the summary of what these alignment techniques are. So dot matrix allows us to visually explore the similarities between sequences, and global alignment compares entire sequences and helps us in recognizing the close or conserved sequences, and whereas local alignment identifies subsequences with the highest similarity and also helps us to detect local evolutionary relationships. So with this, I'll be starting with a quiz. Let me launch the first question. the first question is what is the one element that proteins have but carbohydrates and lipids do not have? So this is the question that we have discussed in the macromolecular section. So let me launch this question. So the first question of the quiz will be the fourth question of your poll. Yes. The question is, what is the one element that proteins have but carbohydrates and lipids do not have? Yes. People are answering in chat also. Yeah, I have received a couple of votes. Let me go ahead. Yes, as everybody mentioned, the correct answer is nitrogen. So let's see from the structural aspects how nitrogen is missing. So we all know proteins, carbohydrates, and lipids are made up of from three basic molecules, that is carbon, hydrogen, oxygen. That is, these three are present in common. So, but when we take a protein structure, we see an amine group. Amine group is nothing but NH2, so which contains a nitrogen over there. And you see all the other three structures, wherever the carbon is common with proteins, the oxygen is common with proteins, and hydrogen as well, but nitrogen is not. So that is why proteins and nucleic acids have nitrogens, whereas carbohydrates and lipids do not. Okay, let me move on to the second question. Second question is, what level of protein structure is primarily influenced by the hydrogen bonding? Or in other terms, this question is what level of structure is mostly stabilized by the hydrogen bonds, or hydrogen bonds form a crucial role in forming these. Yeah, so this second question of our quiz will be the fifth question of your poll. Yes, yes. Yeah, okay, before going on to solution, let me ask, how many of you are facing problems in accessing the polls? They can raise their hand. Because I see only less people answering through polls. Okay, let me go to the solution. Yeah, the solution is a secondary structure. Yeah, I'll give. As I mentioned about, we have discussed about the different levels of the protein structure also. So where the secondary structure refers to the local folded structures that form within the polypeptide chain due to the interactions of the atoms of the backbone. That is, here the backbone is nothing but the polypeptide chain apart from the R groups. The most common types of secondary structures as shown here are alpha helix and beta pleated sheets. Both structures are held in shape by hydrogen bonds which form between the carbonyl O of one amino acid and amino H of other. So when you see when you see here H of one amine group and O of this carboxyl group forms an hydrogen bond to form an helix-like structure. So in an alpha helix, the carbonyl O of one amino acid is hydrogen bonded to the amino H of an amino acid that is 4 down in chain. So what happens is, to make a complete turn and form an hydrogen bond, so the hydrogen bond is formed between the first amino acid and the fourth amino acid. So this pattern of bonding pulls the polypeptide chain into a helical structure that resembles a curled rubber, with each turn of helix containing 3.6 amino acids per turn. The next secondary structure is the beta pleated sheet, where two or more segments of a polypeptide chain line up next to each other, forming a sheet-like structure held together by hydrogen bonds. The hydrogen bond forms between the carbonyl and amino groups of backbone, while this R group extended above or below the plane. So in some cases, the strands of beta pleated sheet may be parallel, that is pointing the same direction, or anti-parallel, pointing the opposite direction. And there is some observance or propensities of these amino acids that are to be more likely or less likely to be found in alpha helices or beta sheets. Can anyone mention which amino acids have this less propensity to be observed in alpha helix? So which amino acid have less propensity to be observed, like less chances to be observed in alpha helix? Okay. The amino acid proline is sometimes called as an helix breaker because its unusual R group, that is, which bonds to the amine group to form a ring-like structure. This creates a bending chain and is not compatible with the helix formation. And in general, proline is typically found in bends and structured regions between the secondary structure. And similarly, amino acids such as tryptophan, tyrosine, and phenylalanine have a larger ring structures in their R group and are often found in beta sheets, perhaps because beta pleated sheet structure provides plenty of space for the side chains. Okay, with this, let's move on to our third question. The third question is, what level of structure that allows hemoglobin to have four identical subunits? Yes, the clue here will be to relate the term subunit where we have discussed the subunit. The question is now available on the polls as well. Okay, I'm repeating the question again. What level of structure allows hemoglobin to have four identical subunits? That means to have four identical polypeptide chains. Or in general, what level of structure that allows a protein to have a different number of subunits, whether they are identical or not, it doesn't matter. Yes. Yeah, the answer is the quaternary structure. So we discussed the concept of subunits here. So these are the four levels of protein structures, starting with the primary structure, followed by the secondary structures, which are local arrangements. Next is the tertiary structure, which is stabilized by non-covalent and other interactions. And finally, leading to the quaternary structure, which includes one or more chain or one or more subunits. As we know, many proteins are made up of only single polypeptide chain. They will end only with the level 3, that is only tertiary level. However, there are some proteins that are made up of multiple polypeptide chain, also known as subunits. So when these subunits come together, they give the protein its quaternary structure. We have already encountered one example with quaternary structure, that is hemoglobin, so which carries blood and is made up of four subunits, two of each alpha and beta types. And other examples are DNA polymerase, an enzyme that synthesizes a new strands of DNA and is composed of 10 subunits. In general, the same types of interactions that contribute to tertiary structure, these are nothing but weak interactions such as hydrogen bonding, Lambda dispersion forces, also hold these subunits together, giving rise to a quaternary structure. Okay, let's move on to the fourth question. So this fourth question, it's you can type this. This won't be available in the poll. So I have mentioned here what is the mutation criteria in normal hemoglobin and hemoglobin that corresponds to the sickle cell anemia. So based on this, so which type of hemoglobin is more soluble in water and which is not? Again, I'm repeating the question. I have provided here what is the mutation that leads to the sickle cell hemoglobin, and when compared with the normal hemoglobin, so which is more polar and soluble in water and which is not more non-polar and not soluble in water. So this we have discussed when a protein or a chain of amino acids is soluble and not soluble when we are discussing about the amino acids. So you can mention for the first question which type of hemoglobin is apparently more polar and soluble in water. So whether it's a normal one or the sickle cell hemoglobin. Yes. Yes. Yeah, the first answer is the normal one. So whether normal one will be the right answer for the second question as well. So it will be more non-polar and insoluble as well. Yeah, second one. Yeah, whoever mentioned the answers are correct. Second one, sickle cell anemia is non-polar because where a charged group is replaced by an hydrophobic amino acid, making it to be more polar and more insoluble in water. Okay, let's move on to our fifth question. The fifth question is regarding proteins which are embedded in outer membrane are called as. Okay. Yes. Everyone mentioned the answer is beta barrel membrane proteins. Okay, let's see how we observe and what is the role of beta barrel proteins. So these beta barrel proteins are a type of transmembrane proteins that are found in outer membranes of the cells, and these proteins have unique barrel-like shape that spans the entire membrane, like as represented in the images, with beta strands forming the sides of the barrel and loops on the outside and inside of the membrane. Coming to the functionality, these proteins, beta barrel, outer membrane beta barrel proteins, are important for variety of cellular processes, including transport of molecules across the cell membrane, signal transduction, and maintenance of the integrity of the outer membrane. Some examples of the outer membrane beta barrel proteins include bacterial porins, which acts as a channels for passive transport of small molecules, and the trimeric autotransporter resins, other ones of which mediate bacterial adherence to the host. Due to their unique structure and functional importance, beta barrel proteins have become a subject of intense study in the field of structural biology and biochemistry. Okay, with this, let me. This is again the question is based on main properties of channel proteins include. So, what are the main properties of channel proteins? Yeah, they helps us access a gates and selective ion conduction. Okay. So coming to the explanation of channel proteins, they are also a type of transmembrane proteins that acts as a pathway for passive transport of ions, small molecules and water across the lipid bilayer into the cell membrane. The main properties of channel proteins include selectivity. That is, channel proteins have specific selectivity for the type of a molecule or ion they transport, allowing them to regulate the movement of specific substances in and out of the cell. The second uh, property is permeability. Channel proteins are highly permeable to the substances they transport, allowing for rapid and efficient flow of ions or small molecules across the membrane. Second thing is regulated gating. These channel proteins also have the ability to open and close in response to the changes in physiological state of the cell, such as changes in the voltage or concentration of specific ions. This allows the cells to control the flow of ions or small molecules across the membrane. Next property is ion specific transport. Uh, so many channel proteins are specific for a single type of ion such as sodium, potassium, calcium, chloride, and can regulate the concentration of these ions within the cell. And other property is structural specificity. The structure of the channel protein is specifically designed to allow the selective transport of some specific ions and molecules, and this is achieved through the arrangement of amino acids in the protein, which forms a pore through which ions or molecules can pass through. And energy dependence. Channel proteins mediate the passive transport, which means that they do not require energy input from cell to function. This makes them an important part of passive transport system, allowing the cell to maintain homeostasis without expending energy. Next question is, what is a comprehensive database for enzymes? I hope this concept is discussed by sir. The videos. Yeah, this is a pretty straightforward question. Answer is Brenda. Okay, let me move on to the next question. One PAM means one accepted point mutation per how many number of residues? Yeah, okay, let me give an explanation to the PAM matrix before moving on to the answer. The PAM matrix, also called as point accepted mutation matrix, is a scoring system used in bioinformatics to quantify the evolutionary distance between two sequences. The PAM matrix is based on the observation that the probability of single amino acid substitution occurring in proteins is proportional to the evolutionary time elapsed since the divergence of two sequences. In a matrix, each cell represents the estimated probability of substitution occurring between a pair of amino acids, and the matrix is usually symmetrical with the diagonal elements representing the probability of same amino acid being represented in both sequences, and the off-diagonal elements representing the probability of substitution occurring. And these matrices are commonly used by protein sequence alignment algorithms to determine the best match between two sequences and to evaluate the evolutionary relationship between them. By comparing the sequences to a reference PAM matrix, it is possible to determine the likelihood of a specific amino acid substitution occur over a given evolutionary time scale. So different PAM matrices have been developed for different evolutionary time scales, with most common one being PAM1, PAM10 and PAM100, which represents the evolutionary distance of 1/10 substitution events per 100 amino acids respectively. One second, there is a question. Yeah, actually the recorded live sessions of week 1 and week 2, we are yet to create a YouTube links. Once these YouTube links are created, NPTEL people will be mailing you, and you can also find them in the problem solving session, the last tab. Like now you are seeing the week 3 tab, right? Under the week 3 tab you will be uploading. Since this is start, so we have some clarifications to get before uploading the link. So we will be doing within this week. We will be getting the recorded sessions of week 1 and week 2. Okay, where am I? Okay, I'm discussing about different PAMs. Okay, now we have a PAM matrix, that's fine, but how these values are derived, how this PAM matrix is filled. The numerical values in PAM matrix are derived from extensive analysis of large dataset of protein sequences and are designed to reflect the observed patterns of evolution in proteins. By using a PAM matrix as reference, it is possible to determine the evolutionary distance between two sequences and to evaluate the likelihood of specific amino acid substitutions occurring over a given evolutionary time scale. As I mentioned, there are different names for this PAM matrix. Okay, PAM10, PAM40 and PAM100. So let me explain them. So these are the different versions of point accepted mutation matrix, which is a scoring system used to quantify the evolutionary distance between two protein sequences. So the numbers here 10, 40, 100 refers to the evolutionary distance, which is expressed as number of expected amino acid substitutions per 100 residues in sequence. For example, if we take PAM10, a PAM10 matrix represents the evolutionary distance of 10 substitutions per 100 residues, which corresponds to a relatively short evolutionary time scale, and this matrix is useful for comparing closely related sequences. Next is PAM40. A PAM40 matrix represents an evolutionary distance of 40 substitutions per 100 residues, which corresponds to a moderate evolutionary time scale, and this matrix is useful for comparing moderately divergent sequences. Next is PAM100. A PAM100 matrix represents an evolutionary distance of 100 substitutions per 100 residues, which corresponds to a longer evolutionary time scale and useful for comparing distantly related sequences. So next, let us move on to the some basic question. Which of the following represents the central dogma of informatics? Yeah, this question has a twist. Which of the following best represents the central dogma of bioinformatics? Okay, I see different answers for this question. A, B, A, B, okay. In general, in biology, the central dogma refers to DNA transcribed to RNA and RNA translation to protein. But in terms of bioinformatics, the understanding of the process of bioinformatics will go to sequence level understanding, and then sequence is compared to the structure, then structural level information is used to get the functional insights. So the answer here will be first one. This is in terms of bioinformatics. In terms of biology, option B will be the correct answer. Okay. So next is the in the system of nomenclature, which of the following enzyme occupies the first one? Okay, the first system or the IC number that we get is for oxidoreductases. Okay, the next question is the central hub for the collection of functional information on proteins with accurate and consistent and rich annotation is. this is the last question for the day. Let me start. Yeah, I have started the Q&A session where you can post your questions. This is the last question for the day. So this question is related to the UniProt. So UniProt KB is made up of two large sequence collections, which is one is manually annotated and reviewed sequences, and other one is automatically annotated and not reviewed. So what are the names of these sequence collections that are available in UniProt? Yeah, I'll be okay. I have received one answer as Swissprat, that's correct. So what is the next one? The answers are Swissprat and TeamBL. Here are some tasks for you for your practice purpose. This is like takeaway task. The first one is in UniProt, how many MOS protein sequences are manually annotated? The second question is how many of these manually annotated protein sequences are associated with PDB? And the third question is map UniProt IDs of above manually created MOS protein sequences with 3D structures to string database and how many string IDs are mapped. And the fourth question is a dot plot for the alignment of human and chicken hemoglobin beta chain and then identify the segments which are same in both sequences. So any doubt? Here is a question. Yeah, the helix structure and the beta street structures are completely dependent on arrangement of the amino acids that we see. And when there's a possibility of having a 1 and 4 hydrogen bond. Last question. Here is the last question. So these are the two lots collections that you can see under the review tab and non-reviewed collections. So Swissprot has a collection of sequences that are manually annotated and considered as a reviewed sequences, whereas TR-MBEL has manually coming to the Q&A question. Yeah, the choice of forming an alpha helix or beta sheet is completely dependent on the arrangement of amino acids and their neighboring amino acid based on their neighboring 1:4, or we have 310 amino acid, 310 helix, pi helix, beta sheets. It's completely dependent on their arrangement. I hope I answered. To explain calculations, yeah, I'll try to add up an example before posting this. Today I don't have my right pad to show you. That's all for today. If someone is having a doubt, kindly use the Q&A or chat. If no doubts, thanks for attending today's session. You are free to leave. For this PAM calculations, I'll put up a calculations once I upload these slides. I don't have a write pad today to write, and so